Yes, so I've just completed my PhD at the University of East Anglia in population genetics and evolutionary biology. And I also have worked at the Earlham Institute, which was also in Norwich, on the Norwich Research Park, where we do a lot of science research, but also provide services to the outside world in sequencing and bioinformatics and training and outreach as well. But today I'll speak a little bit less about, well, hardly any, in fact, about my PhD research on hybridization and evolution, and about how Julia can be used for bioinformatics and computational biology. So, what is bioinformatics or computational biology, first of all, for anybody who's unfamiliar with it? It's the science of using biological data to develop algorithms and relations among various biological systems. Prior to the advent of computational biology, biologists were unable to have access to large amounts of data. So that's a very dry description. Basically, we get chucked a load of data and we're expected to analyze it with computers. Sometimes it's numerical and it's just like collections of counts in the wild. Sometimes it's phenotypic data of traits of different organisms. Sometimes it's measurements from machines. But most of the time when people think of bioinformatics, they think about biological data as in genome sequences. Since the structure of the double helix and the discovery of DNA and the invention of machines that can read that DNA of organisms, biology has become, the floodgates have really opened to a whole influx of data, and we can now process this data to understand biology in whole new ways. And we really do have a lot of data now, and it's only growing. This timeline shows the amount of data that is sequenced in terms of kilobytes per day. And in 1980, when we first did the first DNA sequencing experiments, what's called an S-lab gel, where you would synthesize DNA in a solution with certain radioactive nucleotides, and you'd use a different radioactive nucleotide. So you do 4 experiments, and then you'd run the DNA through a gel using electrophoresis, which moves DNA through a gel with the electric current. The radioactive nucleotides would settle in different places, and then you could image where those bands are, and then you can get the order of the letters in your DNA. Since then, things have become automated, and we can now do high-throughput sequencing. It's a big bioscience business. Illumina was a company that manufacture and sell a lot of machines. We just call them Illumina machines, where they do what's called sequencing by synthesis, where we extract the DNA out of an organism, it's a very painful process, and chop it up into lots of little bits, and of a certain size. Those are called our fragments, and we prepare these DNA libraries. They go into a DNA sequencing machine, which sequence these short little fragments and generate lots and lots of data, which we then have to stitch back together into the complete genome of an organism. And we have to do this for many, many organisms. And it does generate really a tremendous amount of data. At our institute, Earlham Institute, we're lucky to have really, how can I say it without bragging? We've got the best machines. We have some really nice kit. I don't know how to say it without sounding smug. We have machines called the HiSeq 4000s, which they have flow cells in, which my friend who helps the operations in that department tells me they can produce 750 gigabases per run. So if you assume a DNA base sequence takes the same storage space as a byte, then that's 750 gigabytes per run, and they can run 2 at once. And that's for one sample. And these machines are running day in, day out on all the samples that get sent to us. So we really do have a lot of data. and the rate of increase has tracked Moore's law and may even exceed it, because whereas Moore's law, depending on who you believe, may be breaking down because there's only so small you can get, bizarre new ways are being invented all the time to sequence genomes. There are some promising machines being developed by a company at Oxford, for example, the Oxford Nanopore, which, unlike the Illumina machines, which sequence DNA by synthesis, by essentially synthesizing copies of the little fragments you feed the machine, and then that chemistry gives off fluorescent light signals, which are then read by the machine and interpreted. Instead, you just take your entire DNA molecule. It gets fed through very, very small nanochannels, and the electrical disturbances across those channels get read as the different DNA nucleotides get pulled through. So really, I don't know how people think of this stuff. The single molecule sequencing SMART technology, I'm told, uses zero waveguides, which I'm told work because of quantum. I'm a biologist, so I don't know what that means, but apparently that's how it works. Yeah. So what do we want to do? Bioinformatics is a general term, but we want to do lots of things. The sequence analysis, evolution, and phylogenetics are the sort of things I might want to do. People interested in disease might want to construct epidemic models from data. Some people might want to do image analysis of microscope images. Computational biology often includes things like simulation of biological systems. Structure prediction, you might get a linear sequence of protein, and you might want to think, okay, well, how does this linear structure, linear chain of amino acids fold together and form an actual functional biological unit, protein or enzyme? So we want to model this. Gene expression analysis, we can, as well as sequencing, now that we can sequence genomes and DNA, we can also sequence which genes are being turned on or expressed at any given time, so we can get timescales of genes that are active or inactive throughout the lifetime of an organism or during some other biological process. We can improve medicine. We can create personalized medicine for people if we know their genome and we know what their predispositions are to all sorts of conditions. What do we need to do all of this? So for anybody who's unfamiliar with biological data and sort of what exactly a gene or DNA sequence is, I'll get to that in a bit. But if we want to accomplish all these goals, what do we need as biologists? We need efficient scientific computing, and we need, but we also need it to be general purpose. Most biologists, most bioinformaticians are not from a computational or very mathematical background. Most bioinformaticians were either computer scientists who moved into biology, or we are biologists who discovered we quite like computers and we've never set foot in a lab again. We want to do interactive exploratory analysis. We want to run things on highly parallel hardware. At the Norwich Research Park, we're fortunate to have large HPC functionality maintained by a central team, and we have UV3000s, UV2000s, and we have a whole team maintaining these, and another cluster as well, which is maintained 24/7, constantly running people's algorithms. And we also have some things like the Dragon, which I'm told is an FPGA. So some biological analysis programs we've managed to translate into FPGA and have those run really fast. We'd like it to be open source, because if it's proprietary, we can't verify it works, in which case, why should I believe anybody's results if it's not open and I can't verify their algorithm works? And we want to be able to call existing libraries easily, because there's no point in reinventing the wheel. Traditionally, many specific tools were used for this, so you'd script in a shell, you'd parse stuff with Perl, especially the early sort of text formats used to store bioinformatics, to store biological sequence data. We want to do big computations with C++ and Fortran. Genome assembly is a big task, taking all these DNA fragments and stitch them together into a whole comprehensive genome requires a lot of memory and a lot of time. We want to be able to do our stats in something like R and MATLAB, and we'd have to tweak something and then repeat it over and over again. And it's soul-crushing. Why is it soul-crushing? Because you have to know all of these specific languages. You have to stitch them all together with a shell. It's not very reproducible. You get a lot of, well, it worked for me. And if you're a biologist and the first thing you get to your PhD stage or your master stage and you're told, oh, by the way, yeah, you're going to need to learn to program to analyze all this data, or you're going to need to learn how to do stats. And then you're told, oh, but you also need to know Perl. Oh, yeah, and by the way, you need to get really good with a shell. Oh, and everything you're running is just not going to run very fast, so you better learn C. Most biologists don't spend all their time in front of a computer. They're half and half. They spend years in a lab, and then they sit in front of a computer and analyze the data they've collected in the lab, and the lab is incredibly unpredictable. There are protocols you'll do yesterday that worked, and today they don't work because there are reasons. You can have a protocol that worked in a lab at one institute, you take it to another institute and do it in the exact same thing in that lab, and it doesn't work. Genuinely, there are people who are institutionalized because they can do one thing in a lab really well, and the university or department does not let them go because they need them to run that machine. Every time the student runs it, it doesn't work. Things are a little bit better now. I'm told that these were very dark days because I'm not old enough. I'm a youngling. But we have Python now, we have Ruby, we've got BioPython, Bioconductor in R. We still need to use the terminal, of course. But actually, so long as you're not actually trying to create, improve, or understand the software, it's easier, because actually in all these higher level libraries that we use, it's like lots of C and C++, but in an R candy coating or a Python candy coating. So actually, if you want to understand why something was written the way it was, you still have to learn, or you want to create something that works well, you still need to know all of this. Fortran is especially used a lot in environmental modeling still, because there are really old climate models that were written which they do not want to re-implement because it's too hard. So they just use them, and when they break, they panic and ask everybody in the department, including me. I'm in the department at the university. I was in the Department of Environmental Science, so I got asked a lot of stuff about Fortran, which I have no idea about, because they assumed I was an environmental scientist like them. And you have to also learn how to glue the languages together. One of the first packages I wrote was an R package during my PhD, and I wanted to make it run faster. So I thought, okay, spent some time learning C++ and thought, yeah, maybe it's not so bad. Then tried to talk to it with RCPP, and then halfway through got it to work great. Then halfway through, RCPP went up a version and some stuff changed, and I think they had like RCPP exports now instead of doing it some old way, and I thought, I'm not changing it. It all went back to R, and I just vectorized it as much as possible, and then went, I'm not doing that again. I have so many hours in a day, and my supervisor thinks that productivity is not in code, it's only in papers and things, so code doesn't count. This is bad for biologists, it's a bad situation. But we have to learn and keep up with multiple languages. We have to rewrite code, we have to interface them. Parallelization requires more libraries and more coding. Like in R, you have to learn how to use the parallel stuff, all the snow clusters and so on. It's a sad fact that most programs we actually run on the HPC at Norwich Research Park run single-threaded because they're not programmed to use multi-core functionality. It's unfortunate. But all of this leads to stuff being hard to copy, and that's bad science. And actual peer review, I think we can safely say, forget about it. I mean, the odds that I published a paper with an R package, and the odds that they're an expert in any given scientist is an expert in the same area of at the time it was evolutionary genomics, and also understands and wants to read the R code, and then also knows RCPP and C++ to actually read the software, and this was a software note, it's not happening. And some of it's biology's fault. We don't yet treat the development of scientific software with the respect it deserves. It's our assessment is papers and citation factor. Nobody cares whether your repository is maintained really, really well. And this is despite how critical computers and software is in biology now. It's almost like the entire tech revolution is sort of caught us unawares. Suddenly everything's mathematical, and we're like, when did that happen? We were flying outside collecting bugs and things. I'm being hyperbolic, but there's a certain amount of truth in it. I've had PhD students who are in the same office as me and say, whenever something came up, he'd learn the bare minimum to use R, and it'd be by copying an example out of a textbook and then pasting in his own words and then wondering why it didn't work for him. When you have biologists that resistant to learning how to program, when all they want to do is go outside and collect samples again by their own admission, you can't expect them to write stuff in C. It's not happening. And this isn't just my opinion. Mozilla did a code review a while back. It was a collaboration with PLOS Computational Biology, and they had scientists review small snippets of code in Python, R, or Perl, not from packages, just scripts that were part of a bigger research project, supplementary information, this sort of thing. And most of the scientists said it was the first time they'd actually bothered reading any of the code. And it wasn't written for others to use or even understand, and there wasn't any comments or documentation. So enter Julia to save the day. You can write an entire program or workflow in one language. If you want to improve it, you just profile the slow bits and just write better Julia, and it gets nearly as fast as C, right? I mean, we all know this. So this sounds great, and I decided 3 years ago I'd love to be able to do my bioinformatics with this. These were the benchmarks from, I don't need to show you the benchmarks. These have been shown plenty of times before. This was before I knew I was last. So, sorry. The code is familiar to biologists. This looks like Python or MATLAB, of course. So we went, okay, great. We want to create a community for bioinformaticians and biologists who want to learn Julia and use it. So it started as individual projects. Bioseq.jl, which was done by a guy listed at the bottom there, Diego, and phylogenetics.jl, which was started by myself as an attempt to write a phylogenetics library to learn the language. At some point, Richard Smith from Cambridge got in contact with me, and he's really good at getting people to work together and bringing people together, and he brought in Dan Jones and Paulo Castro to form the beginnings of a community and start writing code that would bring together Bioseq, phylogenetics, and loads of other stuff still. So what do we want? We want all the things. We want a well-maintained base infrastructure of very efficient types and algorithms for biology that's suitable for building higher-level packages on top of. A lot of what I do and what I do as my PhD and also in my workplace is write programs that can be applied many, many times, like small bioinformatic apps or one-off scripts, and you want an infrastructure that allows you to come up with those very, very quickly, because when your PhD supervisor says, can you create this software package that detects regions of interest in a genome and then dates them, or by the way, can it also be able to be deployed on a server and have a GUI so you can just drag your files on and everything, and everything starts getting out of control and you're spending more time coding GTK than you are actually working on the analysis part of your program, and it's all about making it nice for other people to run, then you want something that allows you to do all this stuff quickly. We want constant and consistent code review in order to deal with the fact that most code is not reviewed in academic literature. We want resources, and we want to write our biology in code once and to have more people be able to modify it and review it. So our currently maintained, our flagship package is called BioJL, but we also have some others. libz gets quite a bit of attention sometimes because it's just generally useful. And a lot of these others sort of are dependencies of the flagship package. But buffered streams is one that's generally useful as well. Just streaming in information in buffers speeds up things a lot. So BioJL there's one module, bio, and then there are several smaller modules, submodules. You have bioseq for sequence code, bio.philo currently for phylogenetics and evolutionary analysis, and bioalign has lots of algorithms for sequence alignment. And I've just had another one, biovar, which is going to be to do with analyzing biological variation, mutations, and this sort of thing. And the list keeps growing. And I'm going to show you how Julia has been used on the biosequence module, because it's the most mature module out of all of them, and I think it's one where the type system and the parametric type functionality of Julia really shines and allows us to do something that's really interesting that no other bio dot whatever project has been able to do. So biological sequences. A sequence is the primary structure of a nucleotide, of a DNA or protein molecule. So the primary structure is just the sequence of components in linear order. So in the case of a protein molecule, it's the sequence of amino acids that make up that protein. In the case of DNA or RNA, it's the sequence of nucleotides. So why do we care about these sequences? Well, the central, I won't say dogma, of biology, you're taught this at university if you do biology, this is biology 101, is all the living things have DNA in, and it is composed of 4 bases, A, T, C, and G, and it's wrapped into a double helix, and they're complementary. And that's probably where a lot of lay people's knowledge of DNA ends. How exactly it makes a living thing, stuff, magic. But what actually happens is, in a nutshell, groups, the genes in the DNA code for proteins, which are then the building blocks for building an actual squishy organism. So how do you get from the DNA, which is like the blueprint, to the protein? Well, it's read, the DNA molecule is read in sets of 3, which are called codons. So ATC would be read, then GAC, and then TG, whatever comes next. And each unique set of 3 codes for 1 amino acid, and then that forms a unique protein which folds together into a unique structure, and it's an enzyme or a building block or it's part of a chemical pathway or anything. There are some regions that don't code for protein. They're to do with regulation, which genes are being turned into their, which genes are being translated into their protein product at given times. And the idea is you go from a genetic blueprint, and then through the process of development, you go from a single cell to a full-grown squishy human in short order. So that's why we're interested in DNA sequences, and why we're interested in RNA sequences, and why we're interested in protein sequences. So it's a core data type. If you're a biologist and you're not, there's rarely a biologist who has not worked with a DNA sequence. Time for the Julia parametric type system to shine. they're special purpose types rather than simply strings. And the DNA, the alphabet of the alphabet of DNA, A, T, C, and G, is only 4 characters. So a naive script might process a DNA sequence by just treating it as a string or an array of characters. That's wasteful. It's wasteful in memory and it's wasteful in computation time. So we want a type that can compress a sequence. And we came up with a sequence representation which can encode and compress the DNA at different levels. It can encode them with using only 2 bits in the case of a DNA sequence which only has the 4 bases in. But actually, DNA sequencing machines are not perfect. Sometimes they're not quite sure what the letter at a given position is. So it will throw out some other character, or we'll get some other character, like we'll get an N or something, where it means, okay, this might be an A, but it might also be a G, I'm not sure. And the worst case is if you could just get the character N, which is just, I don't know what that is, could be any of them. And we also there are also dash characters, sometimes in biological sequences, where if you have 2 sequences and they get put through an alignment algorithm, so they're lined up so as they have like a high degree of similarity in different regions, and it allows us to infer like evolutionary history, whether a chunk of DNA has been inserted or deleted, if there's been a mutation. Those gaps represent where the alignment program's sort of gone, there's something missing there, or something got inserted there or deleted. Those are called indels. So in that case, you have more than just 4 characters to compress and encode. So you can use a 4-bit encoding, which allows you to add in those extra characters. So how did we do this? First, we had to create the biological symbols. And we did this by creating an abstract type of nucleotide, which was basically just an 8-bit bits type. A DNA nucleotide and RNA nucleotide that both inherit from abstract nucleotide. Because DNA nucleotide is different to RNA nucleotide because RNA nucleotides have a slightly different... one of their bases is not the letter T, DNA thymine. It's called uracil. It's just a quirk of biology. So these are all the different, and you can see we encoded them using 4 bits. And for example, a gap is just all the zeros. A is 1. C is the second bit on, and so on. And then when you get to the ambiguous characters, they're encoded using 1 hot encoding. So if the nucleotide could be an A or a C, then it's both the bits for A and C turned on. So that allows us to do very nice things. You could just use these in an array anyway and just work with these. But next we create types to represent the alphabet that corresponds to those sequences. So we created abstract type alphabet and DNA alphabet and RNA alphabet. Both of those 2 are parametric, where N is the level of encoding. So you can have DNA alphabet 2 or DNA alphabet 4. And the code there is just how you can define your own alphabets. As long as you define certain methods, if you can define the L type and bits of, and you can encode like get to that in a bit. Just for the alphabet, you just need to define these methods. And then we created the biosequence type, which is a parametric sequence type. It has a vector of uint64s, which is just its data store, part, which has a unit range, and then shared rich bool, I'll explain in a minute. You can see we've type aliased it, so as you've got a biosequence of DNA alphabet 4 is just DNA sequence, RNA sequence is a biosequence, parameterized by RNA alphabet 4, and so on. And have I? Okay, I've missed something out. Never mind. These types as long as you encode, as long as you define an encode and a decode method for a given alphabet, Biosequence will be able to store and compress any kind of biological symbol that you come up with. So I create one for storing sequences of mutations soon. So all we have to do is create an alphabet, and then everything in the Biosequence API just works. So it's quite cool. And the part and shared fields are when you want to create a subsequence. What happens is, rather than copying all the data that it needs for that subsequence, rather what will happen is you'll get another Biosequence variable that gets created, but actually it's a pointer to the same vector of uint64s, to the same data in memory, but rather what's different is the part fields are a bit different, and you've got the shared flag bool gets turned on. And then, so subsequences are really, really cheap, which is not something a lot of other bioinformatics libraries do. And then if you ever manipulate one of those subsequences, then it actually goes back to the original data store, copies the data it needs, and then modifies that copy. And all this happens without a biologist having to even know it exists. That behavior just happens. They don't have to care. And we thought that was really important because there's a lot of code like that where biologists, when they're scripting, won't worry about the internal representation, what's going on in the computer's memory or whatever. They will just copy a DNA sequence, or if they slice a DNA sequence, they will just slice it. They won't worry about views, the difference between a slice and a view. Say they won't worry about it. They'll just do the indexing operator in all their scripts. So it was really important to get that subsequence behavior in there. The Biosequence types are really fast. So here's some simple operations. Reverse complement. Biologists will know what that is. Basically, if you read a DNA sequence from the opposite end and translate it into its base complement, the nucleotides that will be on the other strand, that's a reverse complement. Counting k-mers, which is going through the sequence and counting the k-mers that are in there, counting the individual nucleotides, their frequency, and then translation, which is doing that process of taking 3 nucleotides at a time in the DNA sequence, translating it into what would the amino acid be, and then getting the amino acid sequence out. And in all cases, it beats R's biostrings and really beats CCAN in C++, which I was surprised at. I thought C++ was meant to be fast. But I think it just goes to show, like, if you don't write something in a good way, then you're not going to get good results, which is people assume that if you want to write something fast that's bare to the metal, as somebody at work always says, then you have to use C or C++. But no, it is quite possible to write really horrible code in C++. And my experience is that actually since I've started writing Julia, because it's easy to just do code native and code LLVM, I've actually cared a little bit more about those underlying instructions and cared a little bit more about whether those machine instructions are nice or not, and bothered to learn what those assembly symbols mean. The speed is the result of efficient operations on a compressed representation. So because the nucleotides are compressed and squished into these large into these 64-bit integers, you can do operations on either 16 or 32 nucleotides all at once. And that's before you factor in SIMD and all the speeds up of all the other speeds up that Julia just gives you. So that's biosequences. We also want to solve things like this. I hope you find this as amusing, but also kind of scary as I do. This is a FASTA file format. So FASTA is a text file format for storing DNA sequences. It's actually really simple. Every sequence starts with a brace and then a name. And in my sequence A, my sequence 1, whatever. That sequence is slightly more complicated because the user's annotated some things and it's got some numbers in there that mean something to the person whose file it is. And then you have a new line, and then you have the sequence in characters. So pretty simple, right? And seekTK is just a little, it's a command line program, but they're just using it to print out that file. Simple format, right? So that's seekTK output on a file. So clearly the parser's not validating anything. So does anybody want to hazard a guess what this is? Unless if they've seen this before, they might have seen this before. But does anybody know what this file is? It's not a sequence file. Hmm? No, it's not. It looks like brainfuck, doesn't it? I'm not joking. And the parser that is used in seekTK is used 1,615 times in projects on GitHub. And if it doesn't catch that, what really like genuine human errors is it not catching where they've accidentally put, I don't know, a left bracket, they've been going through their sequence file and one of the bases is suddenly an exclamation mark or something else. Yeah, it's not good. Julia on its own as a language can't fix this because it's just bad behavior, but hopefully in this community we can, right? So the solution is don't write parsers by hand. We've decided to use something called Ragel. Daniel Jones deserves an astronomical amount of credit for this because it's almost entirely his. He forked and modified Ragel and then wrote a Julia Ragel package that would allow us to write parsers using a formal specification of what a faster formatted file is, and then you generate really efficient Julia code by running that through Ragel, and Ragel will just generate the code. And actually, you could use that to generate some C code if you wanted, like anything. Like if you can give it to Ragel, you can do it. It's really unfortunate that Colm Networks decided not to support any other languages apart from ASM, C, and C++. Now, but our fork of Ragel still works, so we're okay for now. But we do need to figure out what to do in the future regarding this because we don't, this is a really cool solution. Like Ragel's good; it solves that. So if we can, and it's really fast to boot. So if we can come up with something, a Julia alternative, if we can do this, it'd be great. So what else is coming? Well, as I said, we've got a filing module and a var module. Kenta, who's doing a Google Summer Code project, has just finished SAM and BAM file I/O. So SAM and BAM are like alignment formats, which are highly used. So we're really looking forward to having those. We've got a structure module that Joe has done for us and has done a great job. It's just, have the changes merged? Did they get approved? Good. Today. So, this is, I'm up here talking about for a lot of people. We'd also like to work on learning resources. Maybe I'll be able to do that a bit more throughout my postdoc, and bioinformatics app creation and distribution as well. So it'd be cool if you could just write short bioinformatics apps in Julia that you could then just distribute so people who aren't Julia encoders can just have it on their computer and just run it and it all works great. How to get involved? Well, coding is always appreciated, but documentation and tutorials, if you're a biologist and you want to get to grips with Julia, you can become a maintainer, become a member of our organization if you'd like. Our GitHub organization is there, and we're also on the Git chat room. I think the easiest way is just to come to our chat room and talk to us. And so I'd like to acknowledge everybody in BioJulia, Dan, Richard, Kenta, Joe, everybody who's involved. And at my institute, Professor Van Oosterhout and Dr. Matt Clark, who are my PhD supervisors and employers, respectively. And thank you for you guys for putting on this event. Yeah, thank you.
Yeah, for the parser generation that you're currently using Gradle for,
Yeah.
have you, since you seem to be open to alternatives, have you tried out the Julia wrapper for protocol buffers? Have you tried out protocol buffers?
No, I don't know much about protocol buffers.
Okay, so it's essentially a very similar concept, but it's an implementation that Google came up with, and it's fairly popular. I haven't used the protocol buffer wrapper in Julia myself, but I've heard good things about it from other people, so maybe that's another thing to look into if you are exploring alternatives for that.
That'd be great, because it's a current issue. Like we really haven't come to a consensus what to do about it yet. Kenta once had the idea of, well, maybe we should, in the long term, think of some sort of pure Julia representation. But I think that's just beyond any of our ability, especially mine, and also like the amount of time it would take.
Any more questions? Okay, well, it's fine. Go ahead.